Greetings, everyone. I'm Yanni Van Beem, and on behalf of the GeneBank platform and the Crop Trust, thank you for joining us for today's GROW webinar. With me is Faye Wambua Ludeling, who is co-moderator for this presentation. Just a little housekeeping before we get started. We have everyone on mute to avoid background noises that may distract you from listening to the webinar. If you have any questions or comments during the presentation, please type them into the Q&A box in your Zoom control panel. A recorded version of this webinar, as well as the presentation, will be made available on the GeneBank platform website after this webinar. It is my pleasure today to introduce our speakers from the United States Department of Agriculture. The first presentation will be given by Dr. Clarice Coyne, who has served as curator and geneticist with the USDA Western Regional Plant Introduction Station since 1998. Her research focus is on increasing the utilization and global improvements in plant genetic resource collections through genetic, genomic, and phenotypic characterization. Dr. Coyne's plant genetic resource work was first recognized as 2020 Fellow of American Association for the Advancement of Science and 2019 Fellow of Crop Science Society of America. Congratulations, Clarice, on your awards. Our second speaker today is Dr. Gail Volk, who has been a plant physiologist with the USDA National Laboratory for Genetic Resource Preservation at Fort Collins for 21 years. Her interdisciplinary research program uses molecular data to assess and capture diversity in the USDA's apple collection. She also develops and implements methods to preserve fruit collection diversity in a cryopreserved state. In addition, she is co-coordinating an international effort to develop freely available training materials for plant gene banks. The title of their presentation today is The Critical Role of International Collaborations to Improve Conservation and Utilization of Crop Collections. Welcome, Clarice and Gail. Over to you.
Thank you for that wonderful introduction, Janny. And I'd like to welcome our audience today and thank the CGIAR Gene Bank GROW workshop for the invitation to speak today. The talk is broken up into two presentations. I'll present first and talk focusing on utilization of crop collection, and then turn over the webinar to Dr. Gail Volk to speak on the conservation side of crop collections. So we both work for the National Plant Germplasm System of the United States Department of Agriculture. We're dispersed across the United States in 20 locations. And Gail is located here in Fort Collins, and I'm located in Pullman, Washington. The gene banks are have crop priorities based on the environments for the most part. And you can see we're spread out all the way from subtropical down in Miami, tropical in Puerto Rico and Hawaii, and then up into the northern regions of Geneva, New York, Sturgeon Bay, Wisconsin, and Pullman, Washington. Our distributions of our system, this gives you an idea of our 10-year distribution. And in the last 5 years, the National Plant Germplasm System has sent out 1.2 million samples. And this is broken up between approximately 60% to US collaborators, and the rest are international orders. So to kind of drill down into my portion of the National Plant Germplasm System, I curate the cool season food legume collection. This includes peas, chickpeas, lentils, and faba bean. And the focus of my talk is addressing the issue of how the national and international gene banks can serve to empower the global user community. And the approaches that we're looking at are on the same topics of phenotyping, genotyping, and then some information about our first efforts in high throughput phenotyping and the use of artificial intelligence, primarily machine learning, in understanding our gene banks collections. And then to finish out the talk, I'll present several databases, and really the message here is how the interoperability is going to be key going forward. So this is a wonderful graphic that was put out by Esposito et al., looking at the overall view of the plant genetic resources contributing to breeding. And gene banks, we serve many user communities, not just the breeding community, but today my talk will focus mostly on services to the plant breeding community. And I want to give some specific examples from my program and collaborations on using GWAS and genomic selection of genetic resources. So these two components here, GWAS, genomic selection, and a little introduction on landscape genomics. So I'd like to paraphrase what Dr. Peter Bredding, our national program leader for the National Plant Germplasm System, says that gene banks are centers of innovations. So we're innovations in that when new opportunities are presented, that we can respond quickly. So there's very high interest now in plant-based foods, and my crops are high protein crops, especially the pea, Pisum sativum. So with the increased interest in plant-based foods, in the last 3 years, we've sent out over 23,000 packets of pea seeds. And these are going out to new public and private breeding companies that are responding to the plant-based market. So now that we have a first pea genome reference was published last year, and with a good size SNP set for the pea core, this is turning out to be a good resource looking at pea protein. This is just showing the near infrared unit that we use to measure the protein. So this is our preliminary data after 1 year of field study using primarily the pea core collection. This is the frequency histogram of the range of protein found in this diversity panel. This is a preliminary GWAS. So even after 1 year, we're able to find significant SNPs associated with high pea protein concentration. So how are we going to use this data to increase utilization? So this SNP data set is downloadable directly from Grin Global or from the USDA National Agricultural Library, and then a crop-specific database, Pulse database. And the association mapping panel seed is available from Grin Global. And then this is the publication of the SNP data set. And then for the genotypic data, or phenotypic data, excuse me, the Grin Global, this is the public interface. You can go to the crop descriptors, look for pea protein, and then you can download the entire file using either an Excel or CVS format. Here you can see where the buttons are. And this is a very user-friendly and easy to use. And then for the genotypic data, this will come back later when I talk about databases. Currently, we are able to offer the SNP data sets directly from Grin Global using a hyperlink here to download a VCF or HapMap file. And this is how we're currently offering the SNP data set for the pea core collection. And the reason for the VCF, even though it's a very large file, this can be easily opened up into Tassel. So Tassel is a public software. It's curated, so it's fully functioning and updated. So anyone, anywhere in the world can come to Grin Global, download the phenotype data, download the genotype data, and also order the seed to do their own studies. So I want to give an example of our work we've done with Acarda and USAID on the doing a genotyping of the Acarda Lentil Reference Plus collection. And the plus refers to, it includes some of their heat drought tolerant accessions that they've identified. So this is a set of 176 lines. So we genotype them with using the genotyping by sequencing method. We have a bit over 4,000 high quality Snips. We got early access to the lentil reference genome from Kirsten Bed et al. At University of Saskatchewan. And then we used Acarda historic data. And this is in press in Plant Genetic Resources. This is just a dendrogram showing the relatedness of the accessions. These dendrograms are very useful to gene banks and understanding the diversity in a given set, you know, and we can see within the next 10 years, we'll be able to have this kind of information for entire collections. In the USDA system, currently, the only collection that is completely genotyped with, I believe it's 60K SNP set is the soybean collection. But we can envision that curators are going to have these tools to examine their collections more closely. And then here on the right is the structure plot analysis. And most of these admixed, where you see the two different colors, admixed lines here are primarily breeding lines from Acarda. And then this is the historic data for days to first flower, days to maturity, seeds per pod, and 100 seed weight. And so I wanted to take a break here and just talk a moment about historic data. When we put this, we submitted this manuscript, we got, well, even within our internal review, what is historic data and how is that a valid method to do a genome-wide association study? And so what I wrote in the manuscript and it made it through the peer review is that historically, historic data is using that in a GWAS is relatively new and untested. And I only found one publication on a fruit tree that has been published. But as reviewed by Sally Norton of the Australian Gene Bank, this is an approach, using historic data is an approach that should be used for genome-wide association studies and genomic selection. And the reason is because it's a huge wealth of data. So IPK in Germany has published 2 studies of their historic data on wheat and barley going back decades. So this is material phenotypic data that was collected in the ordinary procedures of a gene bank during regenerations to collect the phenotypic data. So this is a rich source for genetic discovery. And I wanted to quote them here that this historic data, the quote is, elevate them to gene banks to biodigital resource centers. So back to the study we were able to identify some SNPs that are associated with important agronomic traits. And the most interesting one in discussions with Dr. Bett was the day the first flower, because they've done more intensive studies on days to first flower in a larger lentil genome diversity panel. And it looks like the candidate gene is on chromosome 3. So that highlights even with a study with a limited set of the historic data that we used, you can just make correct genetic inferences. So I call this a GWAS to go package. You can order the 176 lentils from Acarda. You can download the SNP data file from the PostDB database. You can collect your own phenotypic traits of interest, conduct the GWAS in Tassel or other GWAS softwares. And this can be done, is possible in any country around the world. Wherever there's internet, they'll be able to conduct this study, this type of study. A second project where we moved into using high throughput phenotyping for our GWAS study was root rot resistance in lentil. So this was the same GBS data set, plus we added the lentil core collection for a total of 326 accessions. It's the same GBS source of the SNP data. This is the dendrogram that you see on the side and the top. And this is a heat map of the kinship matrix. So the high throughput phenotyping for the we did phenotyping in the greenhouse and also in the field. So for the greenhouse, the plants so many days after inoculation with Aphanomyces eutyches, a root rotting organism in lentil, high density, high definition photographs were taken of the roots. And here you can see this is the healthy root down to the almost dead root here. And so they were scored using the traditional method of a visual score of 0 to 5. And then they were also collected the digital images, and so they could do color analysis across the roots. It causes a kind of a honey color to darker brown root rotting. And for the field, the field plot, this is a disease nursery planted in Idaho. And then the drones were flown over on a weekly basis as the disease developed in the field plot. And then this was published in Frontiers in Plant Science. And then in terms of the genetic result, so this was a combination of, besides the diversity panel, there was also a biparental mapping population. And then these are the major QTL regions that were identified in the lentil genome. So moving on to genomic prediction or genomic selection portion of taking genetic resources and doing genomic selection. So this was first termed turbocharging gene banks by Dr. Yu of Iowa State University. And so his idea was, this is a sorghum project, is to use the gene bank collections themselves as the training population, and then use a larger set to do the prediction and validation of the markers discovered in the training selection. So you need the same set of data that you need for the GWAS study. You need your phenotypic data, you need the genotype files. And I wanted to just mention a bit about genomic prediction. We can consider it, you know, was initially discovered or implemented primarily in livestock breeding, and the literature started filling up about 10 years ago. So in just in 2019, someone did this, their Azod et al. Published a review and looked at 91 of these genomic selection publications. And the take-home message here is these are all the different machine learning algorithms that you can use for genomic selection. And then it's a heat map. So these are the most popular. And then they did a comparison just looking at plant height across a number of crops and tree species where they've conducted genomic selections and compared the different machine learning algorithms to see what is getting the best data. And then you can see the algorithms down here, and the higher the number, you can convert these to percentage. So when you get a 63%, that's a really good prediction that you can use in sorghum. So going back to gene banks in our collections and using genomic prediction, I'm working with a professor at North Dakota State University, Nanoy Bandillo, and we're looking at using genome selection in the P-core collection with our historic P seed concentration data. We have a set of about, it's close to 2,000 accessions that have P protein data on it. And additionally, we have other historic data. This is the view from Grain Global, where you can open the descriptor page and look down at all the different descriptors that are available for the USDA P collection. And then I just kind of summarized some of them here. So agronomic features, disease reactions, root traits, not much abiotic, one insect and one nematode. But here, this is in the wrong, it should be over here. But anyway, we have protein, oil, fiber, starch, and mineral nutrients. So that's what will be the traits that we'll be doing the genomic selection on. This is a summary of the total data that's in the phenotypic data for the P collection. And then this is, as I mentioned earlier, the SNP data. And this is just us out in the field. Okay, then these are the kind of questions that we're going to be asking as we go through. And Dr. Bari is the postdoc on this project, and he's has completed these steps to answer these questions about how we can use the genomic selection in the P collection. And we'll be submitting this manuscript soon. Then I wanted to mention about landscape genomics, because this is another powerful tool that's being published on now, where you can take georeferenced accessions and find SNP markers associated with your traits based on the georeferences. So I'll just call attention to this publication. So this is based on high elevation and low elevation. And looking for association analysis across 21 bioclimatic variables. And this isn't exactly a landscape genomics project, but it's looking at the Pisum fulvum, one of the crop wild relatives of pea, is primarily found in a small region in the Middle East. And so a collection of 126 accessions of Dr. Shahal Abo have complete georeference data on them. And so this is overlaying the bioclimatic data. We did GBS Snip genotyping on them. And then this was published recently in Molecular Ecology. And then I'd like to finish up just mentioning about international efforts and databases. So we're fortunate to have the DivSeq organization that includes researchers from across the world. And they have working groups working on genomics, phenomics, and policy. And this fits into how are we going to have standards so that data can be shared among the groups. I think that's the key going forward on the international level. Just mention the GRIN Global Project that, you know, as a community in the United States, we're grappling with how do we go forward on presenting, especially the genotypic data. The phenotypic data is handled well in GRIN Global, but we're working on developing our protocols for now that we have SNP sets that are in the millions. How do we handle that data so that it's accessible? And just a shout out to the GRIN Global people. As a curator, the curator tool has definitely made me a better curator, and it's easy to use, and I have all the information about my collection at my fingertips, a few clicks, and I can answer questions. And also would like to draw attention to the Germinate database. There's a nice recent publication in Crop Science about advances to this Germinate platform, and I would invite people to read this over and go to their platform to see how they're presenting the genomic and phenomic data. And then final to close is to talk about ARS, a USDA effort called Breeding Insight. And so we're excited within the National Plant Germplasm System that there'll be links between this USDA breeding effort and GRIN Global. So I wanted to end up coming back to this nice graphic. And what I like about it is, yes, plant genetic resources are the center of the universe. And with that, I'll close and acknowledge my collaborators on the projects that I mentioned during the presentation. So thank you very much.
Thank you, Clarice. That was a wonderful, comprehensive presentation. You have generated quite a few buzz messages and chat messages about turbocharged biodigital resource centers. So I'm sure we'll be using your terms. Also on landscape genomics and genomic selection, that was great. I'd like to address the questions now. We have 2 questions in the Q&A area before we continue with Gail's presentation. The first question is from Noel Anglin. Is the molecular data, SNPs and reference genome, driving an increased request for distributions, or is it more the interest in plant-based foods?
I think this recent increase on peas is driven by the plant-based foods, yes. But I did see an uptick. We put the Snips out in 2017, and there's not enough time to talk. This is an issue within DivSeq, but which accession is genotyped? So what the choice we made here in Pullman was to make a subset of the original accession so that we had a pure line in order to do the genotyping on it. So that's a set we call the PSP single plant selection. So that selection, that's increased. That set has gone out multiple times to researchers. And so far there's one more PhD that has published on that set. And I know that there's another PhD thesis chapter that's going to be published on it from another group. So the big driver was the plant-based protein. And then another driver is having a genotype set that students can use in their research.
Wonderful, thanks. The second question we have is from Vivian Bernot. How can we make characterization data from our gene bank regenerations more useful as future historic data?
That's an excellent question. And I think it's an issue that I would say, it's just my background. I feel guilty about everything. I think the issue of metadata and community standards is going to be critical, so that when we do 100 seed weight is actually the best one that we have in the cool season food legumes. Everybody, 100 seed weight is 100 seed weight. But then when you talk about plant height or different traits, you know, if as a crop community can, you know, come to an agreement. I'll give an example with the Global Crop Diversity Trust. I'm on the P committee now, working with Mike Ambrose, and our goal is to come up with a standard set for P, because in the past we haven't had that. So if there's within a crop community, and especially if we can on a global basis can come up with defined descriptors, that really helps with the meta-analysis when people are trying to bring data sets together.
Great, thank you. Peter, I was wondering if you want to pose the question live. If we can grant Peter, unmute Peter. He has a question about GWAS analysis.
Okay, yes. Can you hear me?
Yes, we can.
Okay, thank you. Yeah, I'm here waiting for a medical appointment, so I wasn't sure. Thanks. Yeah, I was wondering, when you do your GWAS analysis and you're using accessions that are not necessarily genetic uniform, meaning that you have within accession diversity, how do you deal with aligning genotypic data, which are typically derived from single plants, versus phenotypic data that are obviously then derived from a pool of plants drawn from accessions? So these would be average phenotypic data versus sort of individual genotypic data. How do you statistically align those two data sets? Because I guess that could potentially introduce some noise and reduce the signal that can be detected.
Yes. So on the one study, it was easy because we had pure lines. So the genotype data was collected on a pure line. So on the P study, that was easier. For the Lentil study, it's using the historic data that issue was not addressed. And I think you could see in you know, the p values of the identified Snips were were significantly lower than the ones done on the P study. And then going forward, the study that we have ongoing now, we have that issue. And I think, yeah, I'm going to be working with Professor Bandillo and our statistician here on campus, because yeah, because we collected DNA just on one plant, and our understanding, if we really wanted to represent the genetic diversity of an accession, and they're mostly like land races, so they're definitely heterogeneous mixes, we needed to collect DNA from at least 10 plants, pool that, and that would give us a better representation of that heterogeneity. So I think what it's going to end up doing is, on this current study, is that we're going to we'll have a penalty in terms of what are we going to be able to detect, and what's our confidence level on that, Peter. But I'm hoping that's something that the DIPSEQ, when they come out with recommendations, that they help us with that. Because there's a cost, and that's the thing that ends up happening, a cost with limited resources. What choices are you making, and what are the best choices that we can make? Anyway, thank you.
We have actually one more question, and it's a short one. So let's get this one in. It's from Sadar Sadiki. For GWAS, how much minimum resources or number of accessions would be meaningful in your mind?
Oh, that's an excellent question. And it's interesting now. You can still, what the recommendation is, at least 500. And then, but, people are publishing with, 100 to 200. The larger your diversity panel is, the better result. But then you start multiplying it when you're doing multiple environments, and you have to have replication on any kind of quantitative trait. That completely blows up, and it's very difficult to have the resources to do. For us, it's difficult for me to do an experiment that's over, anywhere from 1,000 to 1,500 plots. It's tough. So that it's really comes down to those kind of resources and the size of the experiments.
Okay. Thank you, Claire. And with that, a virtual applause for you for an excellent presentation. And I'd like to welcome Gail to share her screen and continue the presentation. We'll have more time for questions at the end, so we'll pick up on anything you might have thought about Gail, and then continue with also Claire and then Gail's questions. Thank you.
Great. Thank you for this opportunity, Yanni, as well as the Crop Trust, for letting us speak today. It's a special year for fruits and vegetables, and my research program is primarily working with fruits and vegetables because it's the International Year of Fruits and Vegetables for 2021. So it seems like a particularly relevant time to talk about some of the challenges as well as some of the advantages we have in fruit crop conservation. And I'll be spanning from our work with genetic diversity in apple all the way through to our cryopreservation research. I can't see on my screen. So we have vegetatively propagated collections in the USDA National Plant Germplasm System. And these collections are propagated by grafting or cuttings rather than by seed. They're expensive to maintain because we have to keep them in the fields, in the greenhouses, or in tissue culture. But they have the advantage of having really high economic value. They're important. They add variety to diets, and they provide a lot of job opportunities for local and/or international consumption. These collections offer high levels of novel diversity that are not often available in other gene bank collections. In fact, there's very few international collections that focus on our fruits. Banana is the primary exception. These materials are difficult or impossible to transfer across international and even some state borders, making these accessions that we have in our gene banks irreplaceable and really treasured resources that we need to keep conserved. We also may have limited numbers of associated research and breeding programs. Many of the grains or even some vegetables have many people around the world who are putting forth large projects for these crops. And many of the fruit crops are a lot more limited in their scope. They're also highly susceptible to abiotic and biotic threats, which is dangerous because these things are out in the fields often. They are usually inadequately secured at a secondary location. And we often have fewer technical resources available. We may not have easy propagation methods, especially for our wild relatives. We may not have in vitro methods available or some other techniques that you may have for some different crops. The USDA National Plant Germplasm System has nearly 600,000 accessions, as Claire mentioned, representing over 2,500 different genera. Of these, about 93% are propagated by seeds. There are seed collections. And I'm focusing primarily on these clonal collections, where we have about 69 crops of fruits and nuts, representing around 27,000 accessions. Some vegetables that are clonally propagated with about 2,500 accessions. We have grasses, we have some other clonal crops, as well as about 10,000 ornamental species that are part of our National Arboretum. You can find out more information about these collections by going to our Green Global website that Claire talked about quite a bit. Today, I'm going to be talking about our apple collection. It's maintained in Geneva, New York. There's around 2,500 grafted trees of different cultivars, representing modern cultivars, older historic cultivars, cider apples, crab apples, as well as wild species. There's also over 3,000 seedling trees and crosses, and 1,500 seed accessions from original collection trips. This represents about 35 different apple species and about 15 hybrid species. And a number of years ago, we genotyped the diploid cultivars using 9 SSRs. And it's amazing the longevity of that data set and how it is still very actively used today. I'm just going to touch on a few examples of some of the things we've used this particular data set for, and how in this case, we actually have the advantage over some of the seed crops because one accession is one genotype. And we don't have the variability of the seed lots when we're looking at the specific cultivars because they are clonally propagated. So here's an example of different sets of synonyms that we found in our collections based on these SSRs. And these sets of accessions that are that have the same name, or have different names but the same genotype, are valuable sets of data, especially when you're comparing old historic names and varieties and trying to discuss materials between and among collections. We also identified a number of sport families. These are sets of individuals. In this case, these are all have the same genotype and they're all sports of Golden Delicious apple. We had 23 different sport families in the collection and they're represented by over 100 individuals. And in some cases, we found some materials that are rootstock escapes. So these are things that were actually rootstocks that long ago lost their scions and are mislabeled in the collection as a cultivar, when in fact they are a rootstock. So these SSR data have been actually really useful for us to help understand what the quality of the collection is and how much duplication we have and how we can better explain the content of our collection to our customers. The data have actually been really invaluable for another project that we've been doing for over 10 years now. And we made this data available for genotyping efforts with our national collection as a reference set, so that historic apple trees on public lands or on private lands around the country could be genotyped and compared to our reference set to get cultivar identities. And this is what our first project that we published over 10 years ago, looking at several national parks and ranches down in the southwestern United States and helping them identify the cultivars that they have that are over 100 years old. More recently, just last year, we published some data with Yosemite National Park. And in fact, they're using these cultivar identities that we were able to obtain to figure out how they can conserve their orchards on location. These orchards are usually over 100 years old, and it's really important for them to understand what they could find elsewhere and what is backed up in other places and what they have that's really novel and that they should perhaps be focusing on in their orchard restoration projects. An example of a challenging orchard that they're dealing with is on the right-hand side here. It's an orchard that was actually converted into a parking lot. So they're struggling with how to maintain these old trees and issues with bears visiting the trees in the fall to eat the apples and having cars parked underneath them, and the trees are falling apart. So it's good to be able to understand what we have as cultivars in these collections and how we can possibly protect them, conserve them, and still maintain the activities in the park. Another project that we've done over many years is using this SSR data to understand the diversity of the wild species of some apple. And in this case, we're looking at Malus seversii. It's in Kazakhstan, and it's in the Tian Shan Mountains. We assessed the diversity resulting from the seeds that were brought back from a former curator for forest land trips to Kazakhstan. And we're currently using that information as well as newer SNP data to help work with folks at Caspian University in Almaty, Kazakhstan to better conserve and protect the apple trees of their forests. All this work in apple led to the development of a vulnerability statement, which we published a few years ago, looking at US apple genetic resources. And in this case, we were able to assess what we have in our collection, where the gaps are in our collection, and how we could fill those gaps. What are our vulnerabilities? In some cases, we found some species that were underrepresented or that were represented by accessions that perhaps were not the best representatives or perhaps even hybrids with domestic cultivars. So we've designed and performed a couple exploration trips to augment our collections and help build this diversity that may not be available in any other collections. So this case is collecting Malus sylvestris in Romania and Austria. And then a particularly exciting trip was to Vietnam to collect Malus dumerii. This is extremely poorly represented in gene banks around the world. So our work with vulnerabilities has led to even a larger project, and that was a collaboration with the Crop Trust and Paula Bramall to develop a global strategy to conserve and use apple genetic resources. We published this in 2019, and it's available online. And in this case, we surveyed a number of apple collections around the world. We had 35 collections that responded to our survey, and in these, we've represented over 30,000 or almost 30,000 different accessions of apple in these usually countrywide collections. And it's mostly the cultivars of apple. Our challenge is that apple collections in general are really vulnerable. They're all kept in the field. Many of them don't have adequate funding to support their efforts for conservation. They're vulnerable to diseases and pathogens and environmental threats. There's also a loss in the local diversity because folks are switching more and more to the global production that's dominated by 10 to. We have some crop wild relatives that have some really interesting possibilities, but access to materials and movement of genetic resources is really challenging due to a lot of the quarantine restrictions with countries around the world. And just in general, keeping up these collections are expensive. And we don't necessarily have an international resource like many of the seed crops do. So we proposed in this global conservation strategy for apple that there were some things going forward in the community that could help us improve our collaborations and improve how well we can work together to conserve this important crop. And what we proposed was that we would have an international network of collections that all shared the procedures and came up with standardized procedures for data collection, whether it be genotyping or phenotyping, or even backup standards for their collections. We also proposed that cultivars that are conserved as clones, as cultivars, be located in multiple collections and field sites. So often we have only 1 or 2 trees that are available for a given variety, and it's only present in that one collection in the world. We also thought it was really important to have an information database to document passport, as well as availability, fingerprint information, phenotypes, and images that everyone could have access to, and so that people could share this information on a global scale. And that there were possibilities for conserving seeds for some of the wild species diversity, particularly for folks in collections that have in situ resources within their borders. We wanted to develop a combination of international and local cultivars that are conserved, that represent these unique traits and characteristics, and then find ways that perhaps collections could coordinate amongst themselves to save some of these local cultivars in multiple collections or in multiple locations around the world to provide some sort of safety duplication for them. One thing that's arisen since the publication of this, this global strategy is the use of a 20K apple SNP array. And this has been through the efforts of a number of breeding programs as well as gene bank programs, primarily in Europe. And there's currently over 25 international apple collections that are contributing their DNA and getting the SNPs assessed for their collections. This has provided all sorts of pedigree information. It's providing opportunities to link to markers for breeding programs. It's clarifying synonyms across collections. We're finding in the US collection, it's been very valuable to understand how true our accessions are to species, because in some case we have hybrids between the crop wild relatives and domestic cultivars, and we're actually able to now clarify which species actually is for those accessions, or if they're hybrids. And it's helping clean up cultivar identities. A lot of our cultivars have been passed around the world or passed between countries for centuries. And in the process of doing so, names have been changed. So now we're able to compare the names that we have assigned to trees in our national collections with those in other collections and verify our identities. At this point, we've genotyped over 1,400 of our USDA apple accessions using the SNP array. And it's a continuing work in progress. But the excitement for what this has to offer is pretty tremendous. Now I'd like to go into the second part of my talk, which is about securing clonal collections and how important it is because they're so vulnerable and because they're usually existing as trees in the field. In some cases, we're going to consider a secured collection as a duplicated collection at multiple locations. And that may be the most that can be achieved at this time. In other cases, maybe we can collect seeds from our crop wild relative species and conserve those in a safe, secure place. In other cases, we've backed up some of our collections in reduced temperature in tissue culture, such as in the bags of the strawberry in the middle panel, as well as using cryopreservation. And in cryopreservation, we're storing our genetic resources for the long term in liquid nitrogen. So I'm located at the National Laboratory for Genetic Resources Preservation in Fort Collins. We're part of the National Plant Germplasm System here with USDA. And here at our facility, we have a lot of backup. We have about 82% of the 600,000 accessions that are stored as seed, backed up in the cold vaults and in liquid nitrogen, depending on the crop. And we only have about 15% of our 30,000 clonal fruit, nut, and vegetable accessions. So backing up our clonal accessions is a high priority, but it's also a huge challenge. And that's what a lot of my research focuses on. In the case of cryopreserving materials that are clonally propagated, we generally have 3 choices. We can use dormant buds from cold hardy perennials. And we do this for our apple collection, our sour cherries, willows, and there's some other crops in the research programs. We can also take shoot tips excised from field or greenhouse grown plants. And we've taken these directly without putting them to tissue culture first. And we've done that with citrus and garlic. Or we can take shoot tips excised from tissue culture plants. And we're using this for grapes, strawberry, potato, and actually many others. Here's a listing of what we have backed up in liquid nitrogen at NLGRP. And the methods that we have available for these things are often published, recorded up on our websites. So we've used the shoot tip method for cryopreserving a number of our crops, and we've used the dormant buds for fewer crops, but actually for a tremendous number of accessions. For apple, we have over 2,000 cultivars of apple cryopreserved. And to my knowledge, it might be the largest clonally propagated crop that's cryopreserved in the world. Here's an example about the dormant bud cryopreservation process. So first, budwood is harvested midwinter. So here in the US, that would be January. And it's going to work best if it's harvested from a location that experiences some pretty significant winter temperatures. Once it's harvested, it's desiccated down to about 25 to 30% moisture content and then packaged into tubes. And those tubes are then placed into boxes and they're slow cooled to about minus 30 degrees Celsius and then plunged into liquid nitrogen. We keep them in the vapor phase of liquid nitrogen for the long term. We've got accessions in our collection that have been there for over 30 years. And when we've pulled them out, we still have viable buds. So that's a really promising technology for conserving our crops when we have techniques and resources available. When we want to retrieve them, which in the case of apple, we actually use our cryo collection quite a bit to replenish field trees. The buds are pulled out. A set of 10 in a tube are warmed and they're rehydrated in moist peat moss for a week or two, and then they're grafted onto rootstocks. And then we have a plant that's grafted, it's viable, it's regenerable, and that tree can be planted out in the field. So as I mentioned, we've got over 2,000 apple accessions backed up in liquid nitrogen, and they represent nearly 50 different taxa in our collections. And this is an effort that's been going on here in Fort Collins for a number of years. And just this year, we've started back up to cryopreserve the apple collection some more because we have a number of accessions that need to get replenished, as well as new accessions that have been added to the collection in the past 10 years or so. So we're cryopreserving 100 accessions of apple as we speak. Another technology that we developed for cryopreservation is for citrus. Citrus is super vulnerable here in the US. We have a lot of disease issues. And we were proactive about 10 years ago and developed some methods to cryopreserve it. It was a collaboration between folks that were experts in cleaning the diseases out of tissue culture, or out of citrus plants using tissue culture techniques and micrografting techniques, and our skills in cryopreservation. So in this case, we're taking material directly from this greenhouse, and we're not putting it into tissue culture first. In citrus, it's challenging because plants, every different cultivar or species of citrus would like to have its own media. And we don't have the resources to develop all those media formulations and to optimize them. So in this case, we take fresh budwood that's actively growing from this greenhouse in Riverside. It's shipped to Fort Collins. It's surface sterilized as shown on the left. And then individual shoot tips are excised from the axillary buds. Then those shoot tips are treated with cryoprotectants and plunged into liquid nitrogen for long-term storage. To assess the viability, we take those 1 millimeter shoot tips and micrograft them onto a seedling rootstock. And it's convenient that one single rootstock works for pretty much every citrus cultivar. So we can have rootstocks that are compatible with the scions, and we can assess viability using micrografting techniques. At this time, we've got about 400 citrus accessions that we've cryopreserved and successfully backed up at NLGRP. Another technology that we developed a few years ago is cryopreservation of grape. Grapes are incredibly important. They're grown in collections throughout the world, and they're usually not backed up sufficiently. In this case, we're introducing grapes into tissue culture. We can take nodal sections from those, excise out uniform shoot tips, treat them with cryoprotectants, plunge them into liquid nitrogen, and recover them by plating them onto media. And at this point, we have about 20 of our Vitis cultivars cryopreserved for long-term storage at NLGRP. And a lot of this work was done in collaboration with Giancarlo Spitoni. So there's a lot of challenges in cryopreserving clonal collections. It takes facilities. It takes a lot of specialized skills and training and staff support to make these projects happen. They're labor intensive. We are feeling lucky if we can have 60 to 75 accessions per year per technician for shoot tips for some of these challenging crops for cryopreserving them. For dormant buds, we can process more per technician, as many as 200 or so accessions of dormant buds. But dormant buds are pretty specific as far as which crops we can use them for. It's also, the success of the projects is also dependent upon having sufficient levels of plant material in terms of quantity, qualities, and whether there's endophytes or other contamination issues that we have to deal with. And we're really dependent upon having tissue culture and cryopreservation methods available. So while we're implementing some projects, there's also behind the scenes research going on to get more crops into the pipeline for future cryopreservation efforts. The last part of my talk is going to talk about our training program. And this is some work that we've been doing for the past few years to meet a need in the international community for more resources to train the next generation of plant genetic resource users and managers. This project is funded by a NIFA Higher Education Challenge Grant in collaboration with Colorado State University, Iowa State University, and USDA. And we are developing online training materials that are going to be freely available through a new website that will be associated with GRIN Global. We're making ebooks and ebook chapters, videos, providing images, virtual tours, PDFs, and all sorts of content. And we're also going to have an online class series that's going to be offered through Colorado State University with the first 1 credit module starting in fall of 21. We have a temporary website, which you can see here up on the screen. And this will soon transition over into our GRIN Global website, but that's still in the works. And you can see a number of our collaborators on this project listed below. If you'd like more information about cryopreservation, we actually have quite a bit of information available now, and more will soon be released. And it shows this ebook is freely available. You can go to our temporary website and find the ebook and click on it and find our individual chapters about how we cryopreserve apple dormant buds and citrus and micrograft citrus and also prunus. And we'll soon be releasing our apple and grape cryopreservation methods in this ebook. There's also 2 review papers that we published last year talking about the difficulty and challenges and things to be considering when you're cryopreserving either shoot tips or dormant buds. We have a couple other ebook chapters that are available on our website. One of them is actually a field tour of our Davis collection. It's Mediterranean fruit and nut and grape crops. There's 10 different crops listed or specialized in these chapters. And you can find out, watch videos with the curation staff, as well as read information about these collections. We also have an ebook that we released last year on crop wild relatives and their use in plant breeding. We've got a lot more things behind the scenes that aren't released yet, but they're coming down the pipeline. This includes ebooks relating to plant pathology in gene bank collections. We've got one on fundamentals of plant gene banking, some of the basics, as well as books planned for phenotyping, as well as genomics for gene banks. And some of this phenotyping and genomics for gene banks work will be hopefully done in collaboration with DivSeq, which is the project that Claire mentioned right before my talk. We also have some virtual tours. We have a crop diversity tour, which looks at domestication and diversity in a number of field crops. And that should be released, I'm guessing, sometime this spring. And we're doing a virtual tour for our facility, the National Laboratory for Genetic Resources Preservation, which we hope to come out probably by August. We're doing some more infographics. And this is a preview of what our official website will look like when the time comes. And we're titling it GRIN-U. So in conclusion, the NPGS has a lot of vegetatively propagated collections throughout our gene bank. They're expensive and they're really vulnerable. And we're really dependent upon international collaborations to build these resources, to better understand our resources, to share our technologies and our techniques for preserving and securing them. We need these creative and novel backup strategies to preserve them now, as well as into the future, because at this level of our ability to cryopreserve things, it's really challenging to meet the needs of our system, given the staffing and resources that we have. And all this freely available training content is hopefully going to help educate the current and future gene banking communities, both here in the US and internationally. And I'd just like to acknowledge my group, just barely pre-pandemic when we took this picture, and our staff members who were active in all the aspects of the projects I talked about. And with that, I'll conclude and take any questions.
Thank you, Gail. That was wonderful. We enjoyed, we don't get a chance to listen to people that work on trees that much. We mostly hear people work on seeds. So it's wonderful to know about all your work on apple and citruses and your collaboration with Kazakhstan and Vietnam on bringing some of these accessions in. So that was really nice to see. Also your techniques, very specific on cryopreservation. We have a couple of questions, two or three comments, and then also questions again for Claire. So first of all, from Noel Anglin, let me see, I'm going to the top here. Do you see a big difference in total plant recovery from different tissue sources, like buds, shoots, tips, etc., in cryo? So depending on where you take it, do you see a big difference in the recovery?
We see, so I'll use Grape as an example, and we've taken apical shoot tips and lateral shoot tips and all up and down the stem. In the process of developing our Grape cryo methods, we found that there was a huge range in the recovery levels, depending, and it added so much variation to our results that we decided we had to use nodal sections, because then we could get uniform shoot tips from those buds that are growing from the nodal sections. And that uniformity is what we needed to have so we could optimize our methods. In the beginning, we were taking them from, we were trying to use greenhouse plant material, or we were trying to broaden the project and be more encompassing of where we were selecting buds from. And in fact, it just caused more problems because of the variation. So there's, I think, a huge amount of variation of apical dominance and different maturity of buds that is affecting how well we can freeze things. Mm-hmm. We also are really specific in terms of what buds we take for dormant buds. We want last season's growth. So we don't want old buds from previous year. We're going to take very specific diameters of bud twig material, as well as specific ages of material. So again, we can have a uniform response.
Okay, wonderful, thank you. Benjamin Killian, I'm sorry, did you have anything to add? Okay, Benjamin Killian.
Oh no, I'm just reading the comments.
Yeah, the comment. Benjamin Killian actually posted one of the documents, From Bits to Bytes, Advancements of the Germinate Platform to Support Pre-Breeding Informatics for Crop Wild Relatives. So that's there for you to download if anybody's interested in that one. There's a question from Gul Ali. What are your thoughts on using embryogenic cell suspensions of clonally propagated crops in glycogen and other cryopreservatives for long-term storage in liquid nitrogen or in -8 degrees Celsius? So the question is embryogenic cell suspensions.
Yeah, we, in the clonal universe, we're staying away from cell cultures, and it's because we have a cell-clonal variation concern and a problem. So we try to not have any callus intermediates when we do our cryo, and we would like that shoot tip to regenerate directly from the meristem into its new plant. And having lots of cells in the meristem survive cryo and regenerate that shoot together, just to minimize the chances of cell-clonal variation. We already know that a lot of these crops can easily mutate as a result of our manipulations. And we want to minimize that as much as possible because we are trying to keep very specific genotypes. Right. So it adds an extra step for the case of preservation. It's adding a lot more tissue culture time into each procedure. It may be a possibility for embryos. I'm not usually working with the embryo side.
Okay. There's also another question from Noel Anglin. The collections of new apple genetic resources for Vietnam, for example, were those shared with any other global gene banks and deposited in the USDA collection?
The Vietnamese apples seeds are in our gene bank right now. They have been shared with other botanic gardens in the US, just because we were going to save every tree we had. So we were doing seed germination tests to make sure, to see what our viability was of our original collected seed. And then we had all of our seedling trees, which we genotyped to figure out how much diversity we had. And then we didn't want to lose our trees. So we shared them with one of our repositories. We couldn't put them in Geneva. They're not cold hardy enough to live there. So they went to Corvallis, and Corvallis couldn't keep all the children. So they've actually been distributed to some botanic gardens around the country. So we have seeds available within the gene bank, and we have trees available. We're hoping to grow up some of these trees, and then it'll be easier to distribute and share material when we have more than just our original collected seeds. A lot of the trees we collected from did not have very many fruit, and a lot of those fruits did not have very many seeds. And a lot of the seeds didn't have a high viability level. So we're struggling to really amass enough quantity to share the original seedlets. But hopefully we'll have quite a bit coming down the pipeline as these trees get established.
Yeah, I have a question of my own, actually. Since the signing of Nagoya Protocol in 2014, have you seen that people have opened up a little bit more about transferring genetic resources between countries, or even allowing people to collect and use their resources? You know, Kazakhstan and Vietnam are quite open, but when you collaborate, and this is a presentation about international collaboration, have you seen that the Nagoya Protocol has made a difference? Maybe it's more about personal trust and personal connections, but how do you see this going forward? Do you see a door opening, somewhat more of an open relationship with other countries in the exchange of germplasm?
You know, I don't think I'm the right person to answer that question. Mine are pretty specific efforts that I have with apples in Europe and in Asia, and the countries that I have done these work with have been open, and we're fortunate that some of the species that we're interested in we have access to. In other cases, we have collaborations with countries that aren't able to share germplasm, but they can share DNA. So we've been able to do collection comparisons and genetic analysis and understanding what we have in our collections and their collections on the bioinformatics side, maybe not on the germplasm side yet. But we definitely share our technologies, because having it preserved anywhere is good, and better than having it preserved nowhere. So we are delighted to share all the technologies we have with any country, and hopefully if it can be conserved within their own country borders, maybe the future will open things up a little bit more. It's also really hard in fruit crops because we have so many quarantine restrictions. Seeds are a little easier for the wilds, but if we're trying to move cultivars around, we can import 50 apple and pear cultivars into the US every year. And it goes through the quarantine program, and that could take easily 6 to 8 years before it gets released from our quarantine program. So it's a very slow pipeline to make sure that materials coming into the country are safe and secure. And many other countries have the same problems. So we're not sharing materials to the extent that seeds can be shared. And that makes our ability to share things bioinformatically and genetically using shared SNP arrays and similar phenotyping methods, and that even more critical, because now we can share, we may not be able to share the exact seed set, but we can tell you which seed set in your collection matches, or what cultivar in your collection matches what we have in our collection.
Wonderful, thank you. Michael Aberton has a question. Michael, would you like to pose that question online? Otherwise, I can read it off of, maybe I'll just read it. Do you analyze genetically the relatedness, how close things are, with markers before doing cryobanking for clonal crops? As clonal crop collections may have same accessions with different names or different locations. He's wondering about the duplication. How do you verify that what you're not putting into the bank is already duplicated, or maybe extremely close?
We're definitely having these conversations. And I gave a little webinar with amongst some of our clonal collections about a month ago or a few weeks ago, talking about how we need to be really smart in how we prioritize materials for clonal cryo, because it's so much work. And we have genetic data for collections, and we really should be incorporating that information into our pipeline of the upstream decision-making process of what enters into the cryo pipeline. In the past, that perhaps hasn't been a priority, but more and more looking forward, and now that we have this information available, we're definitely considering it. And it's coming into play as we prioritize apple materials.
Okay, we have 4 more comments if you're okay. Are you still okay? Well, some of them are comments and some of them are questions. We still have 1 more question for Claire, and then we promise we'll end it.
Okay.
Let's see. Same question. Let me see. Where was Tom Payne's question? Gone. Okay. How difficult, related to what I was asking, how difficult to get international germplasm in terms of regulatory and IP issues? I think you kind of addressed that, but perhaps you want to say something else to that question?
I think it's difficult. There's a lot of rules to follow, and I think every country has its own rules. And I think those of us in fruit collections are especially aware of all of the challenges that are out there. But yes, it's a big deal in terms of movement and quarantine and security and safety, and in addition to the political and the IP side of things.
Right. So the comment by Tom Payne is that USDA has funds specifically allocated to support for collecting, with support going bilaterally to various countries. Okay, he did want to make that point. And then Olakunle Sansa, I hope I'm pronouncing that right. Could you please share the link to the training materials? Thank you. So if perhaps somewhere maybe Faye can support us in getting the link to the training materials that Gail is currently working on. She said she did have a page with it's coming in the fall, she said. So we'll link you up to that page of upcoming training materials.
Yeah, and it was at the bottom of a bunch of my slides, and the slides will be out there. Like the whole last set of slides all had that listed as the temporary training site website. And eventually that temporary will link to the real one in a couple months when the real one's complete.
Okay. We have the last question for you would be impact from Dr. Siddiqui. In Pakistan, there is a very scented apple, but its plants outside the habitat lose its traits. How to compare such changing traits in ex situ collections? Wonderful question.
That's a tough question. I don't know. Phenotyping is hard. Our tree fruit crops, and we have the advantage of having single genotypes across cultivars, but we have a huge amount of G by E variation. And retaining certain phenotypes when they're planted in new places or on different rootstocks or whatever the case may be, it can be really challenging. I wish I had answers, but I don't think any.
Yeah, and it goes across multiple crops. Like if potatoes grown in the region, you know, in Peru, Colombia, and Ecuador, you plant them outside of there, they just don't taste the same. And it's the same for almost every single cultural dish that there is. You know, people grow their stuff, and it's just the environment that makes something like that. Very, very good question and very difficult one. Okay, our last question for our speaker today comes from Luigi Guarino, and he would like to know the best way to choose a diversity panel for GWAS. And this is for Claire.
I think the target trait is important. So yeah, so because like a general core collection may not have the specific trait that you're looking at, and I think you know, as databases become more filled with information, it's going to be easier to make selective panel choices. So like I always point out, because people kind of get the mistaken, researchers get the mistaken impression that if I go to the core collection, the diversity of the entire collection is represented. And that's in a relative term. But there's an excellent example in lentil Colletocorum resistance. The entire lentil collection of about 2,500 accessions had to be screened to find three accessions that had some level of resistance to that disease. So that's where we just have to work with all the information that we have. The FIGS approach is very important. And, you know, these approaches of landscape genomics is going to add to it. And just to make life more difficult for the database people, how do we overlay the environment information? Are those always going to, and the reason I ask is as we put in a data set, we go into one environment, we collect the data. We also have the metadata also includes that environment that year. You know, temperature, precipitation, relative humidity. How are we gonna, you know, researchers are gonna need that data. This is a huge challenge, but we're gonna need that information in order to go forward. And yeah, so, you know, for most traits, people essentially just use a diversity panel that's already selected and the core as a core collection as example, but you know, it's not just a worry, it's a fact. You may miss what you're looking for. So I think, you know, the best advice that I give researchers, they go in and say, well, you don't have cold tolerance. How am I gonna, you know, work on this? And I just ask them to please, once they publish, give that data back to us. We'll keep enriching the database, and scientific progress is never, well, unless you're a Nobel Prize, it's not usually a leap. We're making incremental progress. So that's a long-winded answer, but that's kind of my thoughts.
Wonderful. Thank you very much. Well, with that, I think we come to an end. First of all, I'd like to thank my speakers. Again, a virtual applause for both of you for a really, really nice presentations and having the patience to answer all our questions. Thank you to Faye and Linus who have supported us at the Bonn office and made this presentation quite a success. Thank you. And everybody else, the panelists and the people attending, thank you so much. We look forward to seeing you in April. We'll have Fiona Hay diving deep into viability and longevity. So that'll be a pleasure to have her on. And until then, everybody, please be safe and stay in touch. Thank you. Bye-bye. Have a wonderful rest of the day today. Thanks. Bye-bye.